All right. Next up, we have Chance. I'm proud to introduce Chance McGarey. Chance got his BS in Wildlife Ecology and Conservation from the University of Florida. While in college, Chance learned how to band birds at the Prairie Creek Banding Lab and then spent months as an Everglades Biocore Flex intern. But basically, it's an internship of being a Florida man. He maintained box traps for invasives, caught and measured tegus, hacked away at Brazilian pepper, and looked for sea turtle nests, all in the same internship. After graduating, Chance worked on grasshopper sparrows, where he picked up important bird nerd skills like nest searching and boiling ants. Chance is a consummate naturalist and an avid bird watcher, a true rarity in today's pool of intern applicants. He got busy as soon as he got to Archbold and found a field sparrow in the burn that's north of Station. There are only 3 records of that bird from Highlands County. I never got to see it, but it's going to be a minute before I can add that species to my Archbold bird list. Chance was an expert nest searcher and was highly observant of jay behavior. He discovered 2 new scrub jay groups while at Archbold, all based on some keen bird watching. I'm excited for him to present his research today.
Right. Please be sure. Perfect. Thank you, Sahas, for that wonderful introduction. And today I'll be presenting my project, which I've titled Arthropods a la carte, because it's all about what the birds like to eat. And yeah. So the initial ideas behind this project are that birds need proteins for successful reproduction and fledging. Could nutritional quality of their diet have an effect on this? Well, yes, there's been a lot of study on that. It's pretty well known in the greater bird world. However, most of these studies, or all of these studies, sorry, are on supplemental feeding and don't look at the natural diet of these birds, which is the unique aspect of my project. And these are all scrub jay studies, but just in the larger bird world in general, it's a popular point of study. So the questions are, do the relative abundance of arthropods in the territory significantly affect fledging success? And I was using body mass as a proxy for fledging success. Or if that's not true, is fledging success better explained by a variety of other factors that have been seen as significant in affecting the body mass, such as territory size or presence of helpers at the nest? So lucky me, there's a whole boatload of historical data to use on arthropod abundance. And here you can see the data sheet that I used. It's several points scattered all throughout the demography tract of Archbold, which are also the oak posts that are Jay and Joe will be sampling at pretty soon here. Pretty exciting stuff. So you may have seen on the sheet, there's 3 main categories of these arthropods that are supposedly what the jays like to eat the most. That hasn't really been proven, but they're certainly abundant in the scrub. We have Lepidoptera larvae, so just caterpillars, moths. There's Orthoptera, which is mainly grasshoppers, katydids, crickets, that sort of thing. However, at the time of this sampling, it also included things like stick bugs and roaches. It was really the catch-all category. And then Araneae, which is spiders. So I had a bunch of historical data, but it's also historical data taken from a long time ago. And you get interesting things that I sort of had to figure out, like there's a bagworm in this other category, but a bagworm is just a moth larva. So do I put that in Lepidoptera or keep it as other? There's other cool things like all the vertebrate data being cut off the top of the sheets because of the copying process, because we don't actually have the originals. They're locked away in a storage room. Yeah. So I had some vertebrate data up here, but then on a lot of other sheets it'd be missing. So I had no idea if I had a complete set of data for that. So I just excluded it from my study. But I wanted to have it. And you also had mysterious notes scrawled on the sheets in a language no one can read anymore, cursive. No, I eventually did figure out what that particular note said, but it took me a while, which has slowed down the process of digitizing all of these data sheets. And there was a lot of data to go through. This isn't even the sum of it. This is just 3 years, or not even 3 years. This is all of 1989. These sheets that look all ruffled are what I went through for 1990. And then this is March through June 1991. But just that alone took quite a bit of time. So like I said, that's March to June 1989 to 91. That's what I focused on, March to June, just because that's the possible period of time where the scrub jays could have nestlings that they're feeding. It just helped me cut down on this data management. That came out to about 500 data sheets, which is 10% of an estimated 10% of the total data that there is remaining. So there's 4,500 sheets left to go through if you want this whole data set, and that's going to take about 400 hours probably. If you're going at about 5 minutes a sheet, which if I didn't have to slow down and read weird notes and things like that, each sheet took me about 5 minutes, so that's a little bit longer than 5 minutes. So I decided to, for my abundance measurement, lump the different orders into size classes. So this is size class 1, 2, 3, 4. And I did that just because we don't know what the jays prefer to eat, like grasshoppers over caterpillars. We can conjecture about that, but I wasn't sure. So I just assumed that the size class is what matters, and they would prefer maybe a larger size prey item than a smaller one. I took the count data from all the arthropods, and I also got a measure of species diversity through the Shannon index. It goes from 0 to a theoretical upper limit of infinity. Most of my values were, like 0 to 2, though. And basically, a 0 in this index would mean that there's only one species represented for that sampling unit. And then a higher value means that there's more species represented. And then my abundance, I just had my 4 size classes and the counts. And all of this, I had year, month, the territory that was in, and the sampling point ID, which are the oak posts that are out there. So I had 1 sheet per point, and then I could use all those points to get the abundance and diversity per territory for the month that the nestling was measured in. And this is a map here of all of the oak posts or bug posts, whatever you want to call them. And I took GIS shapefiles and I associated points with different territories. And that's how I sort of extrapolated those values for the whole territory. So this is 10 sheets right here. This eBay 89 group has 10 points in it. So it's a fairly well represented territory. However, you have other territories like 6s, which only have 2 points that are trying to represent an entire territory, but that was just a limitation of the data I had. So it may not have captured the full variation in the communities. And then I did this for each year. So I did have a shrinking sample size just because I digitized all of these points, and I started out with 101 territories with nestlings, and then that went down to 61 territories with points that were also in them, and then 28 territories with points sampled and nestlings measured for a total sample size of about 74 nestlings. Which brings me to my results. Ta-da, big reveal. It's a flat relation. Yeah, and that the non-significance could be due to a shrinking sample size. And I've got some more graphs that might lead to that as well, or led me to that conclusion. So this is the species diversity in the arthropods, and then nestling body mass. So you can see there's no effect there. Here's abundance from arthropods of size class 1 and 2, just because those were my most abundant size classes. There were relatively few large insects compared to the small ones, so I didn't have much data to go off of. It is a positive relationship, but is a non-significant positive relationship. So don't take too much stock in that. Although I was excited to see like a line that was actually going up. My significant finding, though, was that arthropod abundance varies with the month. This was significant, but that's not a surprise because, you know, you can see that just going outside with how many mosquitoes you get bit by and things like that. But yeah, it's not surprising that arthropods are seasonally variable. And here are two factors that are supposed to play a significant role in nestling body mass, presence of helpers and the territory size. However, they were non-significant relationships in my study, and I think this could be to an insufficient sample size at what I had. If you take all of the nestlings measured throughout all of the years that we have, you probably would see a significant relationship here. So that's why I think that my sample size could be the problem with my results. And you could see a stronger trend if you digitized all the data. Whether that's worth it for future interns, I'm not sure. They'll have to decide that on their own. So what does this all mean? Well, there's a few things it can mean. Like I said, my sample size could potentially be too small, and you could see a stronger relationship exhibited with more data entry. Or because presence of helpers and territory size in previous studies were found to be significant, those could be stronger effects than just the arthropod abundance or diversity per territory, because even if a territory has a lower abundance or a lower diversity of arthropods, if it has helpers, then that are provisioning the nestling and finding food, it might not matter the difference in abundance or diversity. Or if there's a larger territory, there's more space for them to find arthropods. And the methods used in sampling these arthropods may not have been sufficient. I think they added a lot of variance in the data. Basically, what happened was that people would go out to the scrub and would pretend like they're a scrub jay for an hour in a 10-meter radius, and then just see what they found. And I think those methods leave a lot to the observer and the observer's imagination, just what they think foraging like a scrub jay is like. So perhaps that isn't capturing what's actually out there in the scrub. So what do we do from here? Well, you could digitize the rest of the data, and that is the rest of the data there, and see if you can get a relationship out of that. But that is a lot of work. You could compare the data with suburban sites with heightened pesticide usage and see if there's differences there, because I believe that in neighborhoods where they're spraying for mosquitoes and things like that, there could be a drastic difference. That could be a control for the arthropod abundance. I just thought that'd be an interesting project. And we do have a lot of suburban jay sites that we could do this at. Or you could go back to the drawing board and do the same thing, but with more defined methods that don't leave interpretation up to the sampler to reduce that sampling bias. Well, I'd like to thank all of the interns and staff at Archbold for helping me through my time here. I want to thank Shefali for creating my database that I use to digitize all this data. It definitely helped a lot than me just freewheeling on some Excel. And Bob Curry started on this grant to get all this data in the first place. So I want to thank him for that. Yeah, thank you guys for being here. Yeah.
maybe, and I just missed it, but how did you specifically hone in on those 3 years of data? I know you had to filter out missing data, maybe
Yeah.
that was the most complete, and how did that choice of those 3 years potentially affect?
So I wanted 3 consecutive years. And the first thing I found was the 1989 binder. Excuse me. And because we have nestling body mass data from that year, I just started plugging away at it. And that's how far I got. I would have done, there's about 10 years of consecutive data. I would have done all of that if I could, but it's just how far I got in my internship period. Yes.
So kudos to you for doing this, because Bob Curry never did, so.
I know.
Oh, good job. I actually thought right at the beginning you had an interesting, these are the groups that never produced any young, and these are the ones that did produce youngs of nesting weights. I actually wondered whether, just a very simple question, my first question, whether it would be worth just comparing what was bug availability in territories that never went on to produce young versus that you've got the data. It might be an interesting one to ask. And then the second question was, looking at you're doing counts of bugs, the larvae. It would, I think, a big bug counts for a lot. So could you not do a sort of weighted average so you see what is the volume of those big, or what is the mass of those big bugs that are being delivered to those nestlings rather than just a count?
Right.
I wonder if that actually might tighten up that slope.
Yeah, I thought about doing sort of by biomass and counting heavier as a greater value, but this is just the analysis that I was doing. I was just sort of time limited, but yeah, that was something I was thinking about. That would be great. I might go back,
Just to make you feel somewhat better about your data challenge, when we did the collections data, it was 54,000 specimen data points that we put in a digital database, just to make you feel a wee bit better on all data.
Thank you.